morning everyone, I am Dr. Swati Sharma from DRC, University of Delhi. Today we would be talking about Foundations of Microbiology. As we discussed in the last classes, what is microbiology? It is the study involving microbes. The microbes are the tiny organisms which you cannot see from the naked eyes. So you need to use an instrument called microscope in order to visualize them. So the field of study of those tiny organisms which needs the aid of microscope is called as microbiology. And the foundation of microbiology has been laid by the continuous effort of many scientists. So let us first understand the contribution of the significant personalities or the scientists in the field of microbiology. The first one is Anton Leeuwenhoek. He is also termed as the father of microbiology because he was the one who discovered or who invented the microscope. He used one single eyepiece lens in order to make a microscope, and he was the first one to observe bacteria or protozoa in the droplet of the water. So he termed them animalcules or simple bacteria or protozoa under the eyepiece length. He is also the pioneer of the microbial observation. So he is the one who laid the foundation of the entire unseen world, that is the microbiology. Next we have is Joseph Lister. He is the father of antiseptic surgery. He applied the germ theory to surgery and he introduced the use of carbolic acid for the sterilization. So before him, the instruments which were used for surgeries were not sterilized, and he told that these instruments may cause septic as they have certain microbes attached to them. So it is important to sterilize them before using them for the surgery. And then he introduced the chemical sterilization of the instruments using carbolic acid. This gentleman is Paul Ehrlich. He developed the concept of the chemotherapy. The modern chemotherapy is given by him. He discovered the magic bullet, which is Silvarsin, to treat syphilis, and hence he contributed hugely to the field of immunology. This gentleman is Richard Petrie. Richard Petrie was assistant in Robert Koch's lab, and he was observing Koch when he was inventing the germ theory. So he was very much interested in the field of microbiology. He invented the Petri dish for the microbial culture. So all the Petri dishes that we use today in the cell culture lab or the microbial labs are given by Richard Petrie. He simplified the microbial isolation techniques. Because of the discovery of the Petri dish, it was easier to culture the microorganism on the solid media such as agar plates, and single cell colonies of the bacteria can be obtained. Thus, he not only simplified, he also revolutionized the microbial isolation techniques. This gentleman over the right is Charles Chamberlain. He was one of the collaborators of Louis Pasteur. He developed the Poriclean filters, or in other words you can call them like Chamberlain filters. These are the filters that retain the bacteria or the microbes which are present in the air, resulting in the filtration of the air. He also helped in the discovery of the viruses, because once he developed the filters, he said that the filter can retain the bacteria and other living microbes such as fungi. But there are certain microbes which can still penetrate or pass through these filters because their size is much smaller, and these microbes have potential to cause disease, or you can say they have virulence. So later on, these tiny microorganisms which could pass the filters were termed as viruses. This gentleman here is Edward Jenner. He is also termed as the father of immunology because he developed the concept of the vaccine, and along with the concept he developed the vaccine for the smallpox. He was the first person who observed that the kids which are near to the cows who was infected with the smallpox are immune to the smallpox. So he isolated the extract from the kids who developed the smallpox and injected that extract to the healthy individuals or the healthy kids. And amazingly, those kids who received the small dose of that extract developed the immunity against the smallpox. Hence, this was the concept of vaccination. He said that if you inject the extract of the disease in certain way, in lower concentrations, it can cause the immunity, and these can be termed as vaccination. So he is also the father of vaccination and the father of immunology. He is the famous scientist Louis Pasteur. As you know, his work is significant in discarding the spontaneous generation of the life. He was the one who said that the life comes from the life, not from the non-living material. He was the one who disapproved the spontaneous generation with his famous swan neck flask. He developed an experiment in which he found that in the swan neck flask, there was no bacterial or the microbial growth, because the design of the swan neck is such it does not allow the air to pass through the broth, and the broth was clear even for the prolonged period of time. So he concluded that the life must come from the life. It cannot come from the non-living thing. He was the one who developed the concept of pasteurization. Nowadays we use the milk which is pasteurized. So pasteurization is the instant heating and cooling of the substance. The most common pasteurization technique that we use is in the pasteurization of the milk to get rid of the all microbes. So the pasteurization of the milk is only possible by the ideas given by Louis Pasteur. He was the one who developed the vaccine for anthrax and rabies, which are the fatal disease. So he also contributed to the field of immunology and in the development of the vaccines along with Edward Jenner. He is Robert Koch. He was the lead scientist or the mentor of Petri. He established Koch's postulate. He identified the causative agent for many infectious diseases such as anthrax, tuberculosis or TB, and cholera. He said that these diseases are not because of the deficiency of something, but these diseases occur because of the infection by certain microbes, and these microbes are the causative agent of various disease, and each microbe is distinct from the other microbe in the terms of causing the disease. He also developed pure culture method such as culturing on the agar plates or the Petri plates, and using methods such as streak plating or pouring. So he made it possible to culture. This is Martinus W. Beijerinck. He is the founder of virology. So viruses was the one which were discovered later on. He discovered the viruses in the plant. The first virus to be discovered is tobacco mosaic virus, and he developed the enrichment culture techniques for the viruses. Once the unseen world became seen by the aid of the microbes, the organisms such as bacteria, algae, fungi, they were being identified. But viruses was the last one to be identified because they were much smaller than all those and they were acellular. So it was a huge deal when the viruses were discovered, and this gentleman is the founder of virology. Now he is Sergei Vinogradsky. He is termed as the father of soil microbiology. Even 1 gram of soil can have 1 billion of microbes. So it's crucial to study the soil microbiome. He is the one who discovered chemilithotrophy and nitrogen fixation. Nitrogen fixation is the process of fixing the nitrogen which cannot be uptaken by the plants by a bacteria called rhizobium. So the plants such as leguminous plants, which include your pulses, they have rhodonal roots. The nodes of these roots have a bacteria which is termed as rhizobium. This bacteria aids in the fixation of the nitrogen, and then this fixed nitrogen is available for the plants to be uptaken. So he discovered that concept And he also discovered chemilithotrophy, that is the movement of roots towards a certain chemical. He told that the roots of the plant grow in the search of certain chemicals which are deficient in the plant so that they can be obtained from the soil and the plant can grow better. He is the founder of the microbiology of the soil. He also constructed one column experiment which is also termed as Vinogradsky's column experiment. This gentleman here is Alexander Fleming. He won his Nobel Prize in the field of medicine in 1945 because of the discovery of penicillin. Penicillin is an important antibiotic. Antibiotic is a class of drugs that kill the bacteria. So these medicines are very important in today's world because whenever someone is infected by any bacterial population which is pathogenic, that person is prescribed with certain antibiotics. So he opened the entire era of antibiotics, and today we have many antibiotics which are present such as erythromycin, augmentin and penicillin. So without his contribution, we would still be suffering from many infectious diseases. He is Elie Metchnikoff. He discovered the phenomenon of phagocytosis. Phagocytosis is a phenomena in which the immune cells of the body can eat or engulf the viral particles or the microbial particles. So whenever our body gets infected with any microbe, our immune system has a capacity to kill them by the process termed as phagocytosis. This is body's immune defense mechanism. So the immune cell, they phagocytose the pathological cells or the pathological organism. He is also the father of cellular immunology, and for his great contribution and discoveries, he got the Nobel Prize in 1908. This is Emil von Behring. Emil von Behring discovered the diphtheria antitoxin, and he is also termed as the father of immunology or immunotherapy. He made it possible to use certain antibodies to raise the antigen and use it as the treatment. He also got the Nobel Prize in the field of medicine in 1901, and discovery of diphtheria antitoxin marks a very big stepping stone towards the immunotherapy. Now when we have discussed all the major contributions by the key scientists, now let us move to understand what are the different type of microorganisms. Microorganisms can be of two types. They can either be acellular, which means they do not have any particular cell type such as viruses, prions, viroids, or they can be cellular microorganism, or which can be simply termed as microbes. These are different from the viruses, prion or viroids because they are composed of either one or more cells, whereas viruses, viroids or prions, they are acellular. They do not have any cellular organelles. All they have is the genetic material. But unlike them, the cellular microorganism, they have a defined cellular structure, and also they have a defined genetic material along with the cellular organism. So these tiny organisms are composed of either one or more cells, forming the fundamental building blocks of the life at the smallest scale. The scale is so small that they cannot even be seen by the naked eye. We need to use the microscope in order to visualize them or see them. But they are omnipresent. The meaning of omnipresent is that they are present everywhere even though you cannot see them. They thrive in virtually every environment on the earth, from the deepest ocean to the highest mountain, or even within the bodies of the larger organism. There is not even a single place on the earth where you cannot see microorganisms. We have the heat tolerant microbes even in the Yellowstone National Park hot winds. We have certain microbes in our gut. which can fix vitamin B12 for us. So there is not even a single place on the earth where these microorganisms are not present. Thus, it is of immense importance to study them and to know their role. These cellular microorganisms, they belong to different groups. So it's very important to understand those groups. The term entire microbiome encompasses a vast diverse group which includes bacteria. Bacteria are single-celled prokaryotes and they are known for their diverse metabolic capabilities. They can thrive or survive in variety of environments, but each type of bacteria grow under the specific pH or temperature. Archaea is another group of single-celled prokaryotes. It's similar to bacteria, but they differ chemically in their cell wall structure, and they are often found in the extreme environments. They are distinct from the bacteria in their evolutionary history and their biochemistry. They are more potent than the bacteria, and because of their hard cell wall, they are able to thrive in the extreme environments, and this is what make them distinct from bacteria. The evolution wanted to conserve them, and that's why they were empowered with more peptidoglycans in their cell wall, making them more stern and giving them the attribute to surviving in different conditions. The other categories of microorganisms which are cellular is algae. Algae are the photosynthetic eukaryotes. These can synthesize their own food. They can either be single-celled or the multicellular. They can form they are the basis of aquatic food web, and you must have seen algae. Algae is usually, अगर आपने कोई भी वाटर बॉडी में ग्रीन या हरे रंग की परत, स्लाइमी परत देखी होगी, that is your algae. It's slippery, or in common terms it is also called as kai or moss. तो जो भी aquatic food web है ये उसका primary producer है, and in oceans they contribute to the big biomass of the oceans. The next class of microorganism is the fungi. Fungi are eukaryotic organisms. They include different types such as yeast and molds, and they are known for their role as decomposer and in various symbiotic relationships. The majority of the fungi are your decomposers, but they have other commercial uses as well, such as yeast is used in bakery. Yeast is also used in alcohol production. The next microorganism class is protozoa. Protozoas are diverse single-celled eukaryotic organisms, but they are typically motile. Motile means they can move because they have cilia or flagella, and they are heterotrophic, means they cannot synthesize their own food unlike algae. They often prey on other microbes. They lack any photosynthetic mechanism, so they have to rely on other organisms as their food, and they need to prey on other microbes. So these cellular microorganisms are far from curiosity. They are vital for maintaining the balance of the ecosystem. They play indispensable roles in the nutrient cycle, decomposition, and primary production. Furthermore, they are critical for human health. They influence our digestion, our immunity, and overall well-being. Thus, their unique properties make them invaluable in various industrial applications, from food production to biotechnology and medicine. All the bakery products, they basically rely on the yeast. Yeast is also known as baker's friend because it helps in fermentation of the bakery products. Also, the entire wine industry is dependent upon the microbes or the yeast or lactic acid based bacteria. Nowadays, the sewage treatment is also done by the help of microbes, and microbes were crucial in the development of the first vaccine. Thus, it underlines or underpins the importance of the microbes in various settings and shows how these microbes play the crucial role in the vaccine development, in the industry development, in the cosmetic development, and also in the agriculture in the form of the nitrogen fixation by the rhizobium. So now let us address each class of the microbiology or each class of the microbe one by one. Let us begin with the first most class, which is bacteria. Bacteria are the simplest microbes and they are the ubiquitous prokaryotes. So you guys need to know two terms, prokaryotes and eukaryotes. These two are the type of the cells. Prokaryotes are the simple cells which have only a plasma membrane. They do not have a well-defined nucleus. They do not have well-defined cellular organelles. Thus, they represent the firstmost cell during the evolution, and it shows that bacteria were present on the earth even before humans, because they are the prokaryotes. They are the primary cells which were present, and they are the most well known and abundant type of the microorganism as compared to the others. They are usually single celled organism and they are characterized by the absence of the membrane bound nucleus and other organelles. Thus, they are classified as prokaryotes. Their genetic material is located in a nucleoid region within the cytoplasm. So, their genetic material can be anything. It can be DNA or RNA. Mostly it's DNA, but it's not in the nucleus, whereas it is located in the nucleoid region within the cytoplasm of the cell. Thus, the transcription and the translation in the bacteria, they take place in the same compartment. They exhibit a remarkable variety of the shapes which are the fundamental for their classification. So, we can classify bacteria based upon their shape, such as rods. The bacteria which have rod shape are termed as bacillus because they look like rods. They are usually elongated or cylindrical shapes. Then spheres. Whenever you see circular bacteria such as E. coli, they happen in the form of spheres or coccus. So, जिस भी बैक्टीरिया के बाद कोकस लगा होगा उसका मीनिंग है कि उसका शेप स्फेयर होगा. जिस भी बैक्टीरिया के साइंटिफिक नेम के बाद बेसिलस लिखा होगा, इट मीन्स कि वो एलोंगेटेड या लंबा सिलेंड्रिकल शेप का होगा. और जिस भी बैक्टीरियल के साइंटिफिक नेम में स्पिरम लिखा होगा, इट मीन्स उसका स्पाइरल लाइक फॉर्म होगा. जैसे डीएनए हेलिकल स्पाइरल फॉर्म में होता है, वो भी स्पाइरल फॉर्म में होगा. सो spirals or spirum are either curved or spiral like forms. Then we can also have, so this was the classification of the bacteria based upon their shape, but we can also classify bacteria based upon their functions because they have various ecological and biological roles which are incredibly diverse and often indispensable. So, they can also be classified on their functionality. So, first class is nitrogen fixation bacteria. Not all bacteria can fix nitrogen, but there is a class of bacteria which belong to the genera like rhizobium. They can form a symbiotic relationship with the leguminous plants. Leguminous plants are your pulses which will have root nodules, और जिनकी रूट्स में आप गांठें देखते हो, उन सभी प्लांट्स को लेग्यूमिनस प्लांट्स बोलते हैं. इनको लेग्यूमिनस प्लांट्स इसलिए बोलते हैं क्योंकि इनके रूट नोड्यूल्स में एक बैक्टीरिया ग्रो करता है. उस बैक्टीरिया का नाम है राइजोबियम, और वो बैक्टीरिया एक सिम्बियोटिक रिलेशन बनाता है प्लांट के साथ, जिसमें दोनों का फ़ायदा होता है. बैक्टीरिया को रहने के लिए घर मिला और प्लांट को नाइट्रोजन मिली. तो ये बैक्टीरिया क्या करता है? इट कन्वर्ट्स द एटमॉस्फेरिक नाइट्रोजन इनटू अ यूजेबल फॉर्म फॉर द प्लांट ग्रोथ. इट्स अ क्रिटिकल प्रोसेस फॉर द सॉइल फर्टिलिटी. प्लांट को ग्रो करने के लिए सनलाइट के अलावा कुछ न्यूट्रिएंट्स भी चाहिए होते हैं, जिसमें से नाइट्रोजन सबसे इम्पॉर्टेंट न्यूट्रिएंट है. बट बहुत से प्लांट जो भी एटमॉस्फेयर में नाइट्रोजन प्रेजेंट है उसको नहीं ले पाते. तो जो ये बैक्टीरियाज है राइजोबियम, ये क्या करते हैं? ये उन प्लांट्स की हेल्प करते हैं. ये उनकी रूट्स में ग्रो करते हैं और उनके लिए जो भी एटमॉस्फेयर में नाइट्रोजन प्रेजेंट है उसको फिक्स करते हैं और उसको अवेलेबल करवाते हैं प्लांट को, ताकि वो प्लांट उस नाइट्रोजन को ले सके और अच्छे से ग्रो कर सके। तो ये प्रोसेस बहुत इम्पॉर्टेंट होती है प्लांट की ग्रोथ के लिए और उस मिट्टी को उपजाऊ बनाने के लिए। सो विद दिस कंसेप्ट यू विल लर्न हाउ इम्पॉर्टेंट इट इज़ टू हैव गुड बैक्टीरियाज इन द सॉइल। दे आर ऑल्सो इम्पॉर्टेंट इन गट हेल्थ। गट मीन्स योर इंटेस्टाइन, जो भी हमारी डाइजेशन की प्रोसेस है वो गट में होती है। तो बहुत सारी स्पीशीज़ जैसे ई कोलाई हम लोगों की गट में है। सो मैनी स्पीशीज़ सच एज strains of E. coli and lactobacillus, they reside in the human gut. They aid in the digestion and vitamin synthesis and modulate the immune system. So आप लोगों ने सुना होगा कि हम लोगों में पुराने ज़माने में बोलते थे कि जैसा खाओ अन्न वैसा हो मन, क्योंकि जो भी आप खाओगे वो डाइजेशन के द्वारा आपके गट में एब्जॉर्ब होगा। और गट में कैसे एब्जॉर्ब होगा, वहाँ बहुत तरीके के माइक्रो ऑर्गेनिज्म होते हैं, जिनमें से दो सबसे कॉमन माइक्रो ऑर्गेनिज्म है ई कोलाई और लैक्टोबैसिलस। जो आप दही खाते हैं उस दही में भी लैक्टोबैसिलस होता है। अगर आप कभी भी बाज़ार से दही खरीदें, आप उस पर पढ़ सकते हैं, उसके लेबल पे उस पर लिखा होगा लैक्टोबैसिलस कल्चर। तो ये बैक्टीरिया हमारे गट में क्या करते हैं, यह बैक्टीरिया हमारे गट में हमने जो भी खाना खाया है उसको पचाने में मदद करते हैं। और साथ में ही कुछ विटामिन्स हैं जो हमारी बॉडी नहीं बना सकती। तो वो विटामिन्स जैसे कि विटामिन B12 एंड एंटायर फैमिली ऑफ विटामिन B कॉम्प्लेक्स, ये सब इन बैक्टीरियाज के द्वारा हमारे गट में बनाया जाता है और हमारी बॉडी को मिलता है। दे ऑल्सो हेल्प इन द इम्यून सिस्टम। अगर हमें कोई भी इन्फेक्शन हुआ है तो ये गट बैक्टीरिया हमें उस इन्फेक्शन से बचाने की कोशिश करता है। इसलिए ही एंटीबायोटिक्स कम से कम खाने चाहिए, क्योंकि एंटीबायोटिक्स ना सिर्फ डिजीज़ कॉजिंग बैक्टीरिया को मारता है, वो आपके गट माइक्रोबायोटा से मेरा मतलब है आपके गट में जितने भी अच्छे बैक्टीरिया हैं उनको मारता है, जो कि हमारे लिए अच्छा नहीं है। इसलिए हमें कम से कम एंटीबायोटिक का प्रयोग करना चाहिए और सिर्फ डॉक्टर की सलाह पे ही एंटीबायोटिक लेनी चाहिए। आगे चलते हैं कि और क्या काम है बैक्टीरिया के। टिल नाउ वी हैव स्टडीड ओनली द पॉजिटिव साइड ऑफ द बैक्टीरिया, that they aid us in nitrogen fixation, that they help in gut health, but there are also the dark side of these bacteria. They can be disease agents. While most of the bacteria are beneficial, but some are pathogenic. By pathogenic I means they can cause disease. So the disease causing bacteria such as Streptococcus species. Streptococcus species अगर आप पिछली स्लाइड्स को ध्यान में रखते हुए समझेंगे तो स्ट्रेप्टोकोकस हमेशा कैसी होगी, स्फेरिकल, क्योंकि स्ट्रेप्टो के नाम के पीछे क्या आ रहा है, कोकस। तो ये बैक्टीरिया आपको माइक्रोस्कोप में कैसा दिखेगा, ये आपको स्फेरिकल दिखेगा। और ये क्या करता है, ये कॉज करता है स्ट्रेप थ्रोट या गले की खराश। देन अदर बैक्टीरियाज आर ट्यूबरक्लोसिस। ट्यूबरक्लोसिस क्या कॉज करता है, टीबी। दे आर डिफरेंट टाइप्स ऑफ टीबी। मेजॉरिटी ऑफ इंडियन पॉपुलेशन इज़ द कैरियर ऑफ टीबी। कैरियर से मेरा मतलब है कि आप लोग टीबी का बैक्टीरिया अपनी बॉडी में कैरी करते हो, बट उसका डिजीज़ रिफ्लेक्ट नहीं होता, पर हम किसी और को इन्फेक्ट कर सकते हैं। टीबी मेजॉरिटी ऑफ केसेस में सिर्फ लंग्स को इम्पैक्ट करता है, बट ये बाकी ऑर्गन्स को भी इम्पैक्ट कर सकता है। और जो इन्फेक्शियस डिजीज़ हैं बैक्टीरिया से होने वाले, उनमें से एक और सबसे कॉमन है निमोनिया। यूजुअली निमोनिया वायरल इन्फेक्शन से होता है, बट जैसे ही बॉडी को वायरल इन्फेक्शन होता है बॉडी का इम्यून सिस्टम वीक हो जाता है। तो जैसे ही इम्यून सिस्टम वीक हुआ बॉडी पे बैक्टीरिया अटैक करता है और बैक्टीरिया के अटैक से निमोनिया होने के चांसेस होते हैं। सो दिस बैक्टीरियाज आर primarily reproduce asexually through binary fission, where a single cell divides into two identical daughter cells. Thus, it enables rapid population growth under favorable conditions. Many motile bacteria use whip-like appendages, which is called flagella, for their movement, propelling themselves through liquid environments. towards nutrient source or away from toxins. तो इसी के साथ हम बैक्टीरियाज को पढ़ चुके हैं, अब हम आगे पे मूव करेंगे। धन्यवाद।